replication. Hope I didn't scare you there. We left off today with the understanding of leading and lagging strand. Leading, the whole idea that it's called leading because it starts right as soon as helicase unzips and it works in the same direction as it's moving. Lagging strand is going to be the strand that produces the DNA in the opposite direction as helicase is moving. We also talked about these Okazaki fragments. Those are the small segments on the lagging strand that will have to be built in small segments because of the fact that it's working against the helicase. So as a practice, we kind of did this. If I chose that here is my 5' end on the old strand, and I asked you, let's see if you can identify the leading strand from this. Which of these two orange lines would be, let me move the cursor, which one would be the leading strand? So I'll give you 10 seconds to think about that. Let's see if you choose the one down here, you actually are correct. Leading strand. So if this is the 5', this old strand portion will be the 3'. Opposite it, 5', then the inside is 3, so it's pointing towards the 3, which is going the same direction as the helicase is opening. If you flip it around and I do another example, and I say, this is my 3', and let's change the color. Here are the new strands coming in. Which one would be the lagging strand? Lagging. Did you choose this one? Good. 3 down here, 5 on the original, 3 there. It's pointing towards the 3. I can kind of figure that. With the new one, it's going away from the helicase. So just be aware of what I'm asking. Leading or lagging, location of helicase, and the 5' and 3'. To get to where we talked about today, there are some other things. So if you go, I can't remember specifically what class had each one. So these are some of the things. So here's an example of what we did in class with one of the classes. With the 3' being here, this is the leading strand. It goes in one motion. Easy. This one is the one that has the Okazaki fragments. Those set fragments of about 50 to 100 nucleotides, even sometimes 150 nucleotides, some books will say, where you start a little bit in and you work backwards. So today's lesson is really going to be about, there's a little bit more detail that I didn't talk about, but understanding this is more than half the battle. It's more along the lines of there's other enzymes involved. We've talked about helicase so far, which is right here, and DNA polymerase. Technically, there are 2 types of DNA polymerase. The first one, which is really the premier polymerase, is not, there's actually varieties of it. The one that does the majority of the work is called DNA Polymerase 3. That is the one that adds pretty much the entire leading strand and is also responsible for producing all of these little Okazaki fragments. That's the main contributor to building DNA. Polymerase 3. If I compare this to painting a room, before you actually lay down the true paint for a room, sometimes you add what is called, now up-to-date things actually put it right into the paint and mix it in. It's called a primer. With DNA replication, there actually needs to be an enzyme that lays down the original sequence of DNA. It's actually called RNA primase. Now it's RNA. We'll get to what RNA does. We have talked about it a little bit. But what technically happens is RNA primase is an enzyme that will come in and it adds right at the beginning. Let me change it. That's a purple color, so green. Technically, the first few nucleotides that are added are really a group of RNA nucleotides. It's a primer, and it basically tells the DNA, I'm ready to start right here and start adding. It attracts the DNA polymerase 3 to say, I'm ready to start adding nucleotides. So if I were to do this technically, I'm going to redo the diagram. I'll show you what would, so these are all from classes. So this is my DNA that I was technically now going to replicate. We've been working with the one that had 3' up here. So this is my leading strand. Technically, RNA primase adds a short segment of RNA. Then my DNA polymerase 3 comes in and will add, from this point on, in the same direction as helicase is moving, the rest of my leading strand. Not an issue. The lagging strand needs more of these little primers because every time an Okazaki fragment starts, they've got to put a small primer down. Because technically the DNA polymerase 3 can't attach itself to nothing. It attaches itself to the primer that has been laid down. The primase is the enzyme that adds the nucleotides. This short segment of nucleotides is called the primer because it's what is going to be the attachment site of the rest of the nucleotides. On my lagging strand, the first thing that happens is my RNA primase is going to add a short segment, and then my DNA polymerase 3 is going to come in and work backwards. Then the primase goes down a little bit more. The DNA polymerase 3 adds nucleotides up until the last primer there. So this continues for every Okazaki fragment. The DNA polymerase 3 once again comes in and adds to the last primer. Every primer is added by primase. The primer is a small, and I mean we could literally talk about 5 to 10 nucleotides in length. They're made of RNA nucleotides, slightly different than DNA. So this would occur the entire length of my lagging strand. So I'm going to stop right here after this one to say, let's say there were only 4 Okazaki fragments I'm working with right now. This is obviously not going to be a complete DNA. We don't want to leave it like that. So here's what happens. The other DNA polymerase, which is called Dna Polymerase 1. I don't know why the numbering, don't ask me, I'm not sure of it. So the first one will actually come in and remove its job. Once all the Okazaki fragments have been laid, from start to finish, this enzyme goes in and removes all of the little green portions, which remember are the RNA nucleotides. So the DNA polymerase 1 comes in and will change this green segment. It changes it now to blue because it lays down, it changes the RNA to DNA. So we now get rid of those RNA, those 5 or 10 RNA nucleotides have now been removed, and in their place, we don't leave a gap there, in their place, DNA is added, and that little segment of DNA is added by DNA polymerase 1. So then it goes up to the next primer. It says, get out of here, your RNA. Now the rest filled in with DNA. Does the same thing. Gets rid of the green and replaces it with the blue. Gets rid of the green, replaces it with the blue. So we are now a complete DNA strand. But technically there are still little gaps. I didn't really draw them as clearly, you probably can't see the detail, but they truly are still Okazaki fragments. We haven't completely bonded every Okazaki fragment. That is the role of the last enzyme in the process. It's called ligase. You see all the ASE endings, enzymes. Ligase's responsibility, it's just a finisher. It goes in and bonds all of the Okazaki fragments together. So it will look like one long piece of DNA. It resembles now the leading strand. It's one strand, all completed. So you really technically can't tell, in the end, that the lagging strand was made in fragments because of the DNA polymerase 1 helping out, the ligase helping out. What you will get is one long strand of newly synthesized DNA, one long strand of newly synthesized strand up here. We still do have to get rid of this primer. It was originally laid down, so DNA polymerase 1 will act up here as well one time. It gets rid of that primer and adds it there. Whereas on the lagging strand, it was just a repeating of the DNA polymerase 1 getting rid of all the primers that the RNA primase laid down, and then the ligase acting as the glue that puts all of the Okazaki fragments together. So if I continue this, in the end, if I let the helicase go to the end, what you would get is, and I'll change it, imagine the next slide,